Welcome to Sleepy Science Odyssey. Tonight, the brain, the 3-pound organ inside your skull that makes you who you are, processing sensory information from the world around you, storing memories that span your lifetime, generating emotions that color your experience, controlling every movement you make, and somehow producing the conscious awareness that you're reading these words right now. We'll move slowly through the anatomy and physiology, exploring the structures that regulate basic functions, the networks that enable complex thought, the chemicals that modulate mood and behavior, and the mysterious processes that create consciousness itself. If you're new here, this channel is for people who need something to think about before they can stop thinking. Real neuroscience, no speculation, just quiet facts in the dark. Let's begin. The human brain weighs approximately 1.4 kilograms, about 2% of total body weight. Despite its relatively small mass, the brain consumes a disproportionate amount of the body's resources. At rest, the brain uses about 20% of the body's total energy consumption, roughly 320 calories per day. Most of this energy doesn't go into conscious thinking. It's required to maintain the basic infrastructure of neurons and synapses, preserving the electrical gradients across cell membranes that allow neurons to fire, constantly synthesizing and breaking down neurotransmitters, and maintaining the vast network of connections that makes cognition possible. The brain is about 73% water, with the remaining mass consisting of fats, proteins, carbohydrates, and salts. The high water content is why dehydration affects cognitive function. Even mild dehydration, losing just 2% of body water, impairs attention, memory, and mood. The brain's composition changes throughout development. At birth, the human brain is about 30% of its adult weight. It grows rapidly during childhood, reaching about 80% of adult size by age 2 and 95% by age 6. Most of this growth isn't from adding new neurons. You're born with most of the neurons you'll ever have, possibly 200 billion at birth, but from neurons growing, making connections with other neurons, and developing the myelin insulation around axons that speeds signal transmission. After childhood, there's actually a reduction in neuron number through a process called synaptic pruning, where unused connections are eliminated and frequently used connections are strengthened. This refinement continues through adolescence and into the early 20s, particularly in the prefrontal cortex, the brain region involved in planning, impulse control, and decision-making. The adult brain contains approximately 86 billion neurons and roughly the same number of glial cells, supporting cells that were once thought to merely hold neurons in place, but are now known to perform essential functions, including providing nutrients, removing waste, forming myelin, and modulating neural activity. The ratio of glial cells to neurons increases in more complex brains, suggesting glial cells contribute to cognitive sophistication. Individual variation in brain size exists, with adult human brains ranging from about 1,100 to 1,700 grams. But within this normal range, size doesn't correlate with intelligence. What matters more is the organization, connectivity, and efficiency of neural networks. Einstein's brain, famously preserved after his death, was actually slightly smaller than average, though certain regions, particularly the parietal lobes involved in mathematical and spatial reasoning, showed unusual features. The brain size relative to body size varies across species. Humans have large brains for their body size, though not the largest in absolute terms. Elephant and whale brains are larger. The encephalization quotient, a measure of brain size relative to expected brain size for a given body size, is highest in humans, followed by other primates, dolphins, and elephants. This measure correlates roughly with cognitive abilities across species, though the relationship is complex and influenced by many factors including brain structure, neuron density, and metabolic constraints. The evolutionary expansion of the human brain, particularly the neocortex, occurred relatively recently, primarily over the past 2–3 million years. This expansion required dietary and social changes to support the metabolically expensive brain tissue, and it may have been driven by selective pressures related to social complexity, tool use, language, and cultural transmission of knowledge. The brain is the most complex object in the known universe. This isn't hyperbole. By any measure of complexity, the brain exceeds everything else we've encountered or created. Each of the 86 billion neurons can connect to thousands of other neurons through synapses, creating a network of approximately 100 trillion connections. If you tried to count these connections at 1 per second, it would take you over 3 million years. The number of possible neural configurations, the different patterns of which neurons are connected to which, and how strongly, exceeds the number of atoms in the observable universe. This vast connectivity space allows the brain to represent an essentially unlimited range of thoughts, memories, and perceptions. The brain processes information in parallel across multiple systems. When you see a face, different brain regions simultaneously process its color, shape, motion, emotional expression, and identity. These separate streams of information are somehow bound together into a unified conscious experience, a puzzle called the binding problem that neuroscientists still don't fully understand. Visual processing alone involves over 30 distinct brain areas, each specialized for different aspects of vision. The brain operates on multiple timescales simultaneously. Neurons fire in milliseconds. Neurotransmitters are released and cleared from synapses in milliseconds to seconds. Hormones modulate brain activity over minutes to hours. Learning and memory formation occur over seconds to days. Brain development unfolds over years to decades. The same neural circuits that process information in milliseconds are shaped by experiences across entire lifetimes. The brain is also remarkably efficient. Despite its high energy consumption relative to body size, the brain operates on about 20 watts, roughly the power consumption of a dim light bulb. The fastest supercomputers attempting to simulate even a small portion of brain activity require megawatts of power and rooms full of hardware. Part of the brain's efficiency comes from its architecture. Neurons process information locally and in parallel, rather than shuffling information back and forth between separate memory and processing units, as digital computers do. Another source of efficiency is the use of chemical signaling alongside electrical signaling, allowing complex modulation of information processing with minimal energy expenditure. The brain's complexity emerges not just from the number of neurons, but from the principles of organization. Neurons are organized into layers, columns, and nuclei. These local circuits are connected into larger networks. Networks are organized into systems: motor system, visual system, limbic system. These systems interact, creating emergent properties that can't be understood by studying individual components in isolation. Consciousness, creativity, language, abstract reasoning. These higher-level functions emerge from the orchestrated activity of billions of neurons operating according to relatively simple rules, combining in intricate patterns that produce phenomenal complexity. the hierarchical organization of the brain, with information processed at multiple levels from simple features to complex abstractions, allows for sophisticated cognition while maintaining efficiency. Lower levels handle routine processing automatically, freeing higher levels for novel problems and abstract thought. Studying brain complexity requires multiple approaches at different scales. Molecular neuroscience examines individual proteins, genes, and biochemical pathways. Cellular neuroscience studies individual neurons and synapses. Systems neuroscience investigates how brain regions and networks function. Cognitive neuroscience links brain activity to mental processes and behavior. Each level of analysis provides insights, but understanding how these levels relate to each other, how molecules give rise to thoughts, how neural firing patterns create consciousness, remains one of science's greatest challenges. The neuron is the basic functional unit of the nervous system. Neurons are specialized cells designed for rapid communication. A typical neuron has several key components. The cell body, or soma, contains the nucleus and most of the cellular machinery for maintaining the cell. Dendrites are branching extensions that receive signals from other neurons. A single neuron may have thousands of dendrites, creating an intricate tree-like structure. The axon is a single long extension that transmits signals to other neurons. Axons can be quite long. The axons of motor neurons that control your foot extend from your spinal cord down your leg, over a meter in some people. The axon terminal at the end of the axon forms synapses with other neurons, where communication occurs. Neurons are incredibly diverse in their morphology and function. Pyramidal neurons in the cortex have characteristic triangular cell bodies and extensive dendritic trees. Purkinje neurons in the cerebellum have elaborate, flat dendritic arbors resembling fans. Basket cells wrap around other neuron cell bodies. Different neuron types have different firing patterns, respond to different inputs, and connect to different targets, creating the diversity needed for complex brain function. Neurons communicate through electrical signals called action potentials. At rest, a neuron maintains an electrical potential across its membrane of about -70 millivolts, with the inside negative relative to the outside. This resting potential is maintained by ion pumps that actively transport sodium ions out of the cell and potassium ions in, using ATP as energy. The membrane is selectively permeable. Potassium can leak out more easily than sodium can leak in, contributing to the negative interior. When a neuron receives sufficient excitatory input, voltage-gated sodium channels in the membrane open. Sodium rushes into the cell, driven by both concentration gradient and electrical gradient. This influx of positive charge depolarizes the membrane. If depolarization reaches a threshold of about -55 millivolts, more sodium channels open, causing more depolarization, triggering even more channels, a positive feedback loop that rapidly drives the membrane potential to about +40 millivolts. This rapid swing from -70 to +40 millivolts is the action potential. The action potential is brief. Within a millisecond, sodium channels inactivate and potassium channels open, allowing potassium to flow out of the cell, restoring the negative membrane potential. For a brief moment, the membrane is hyperpolarized, more negative than the resting potential, which makes the neuron less likely to fire again immediately. This is called the refractory period. The action potential is all or nothing. Once threshold is reached, the action potential always has the same magnitude. Neurons encode information not in the size of action potentials, but in their timing and frequency. A neuron can fire anywhere from a few times per second to hundreds of times per second. Different firing patterns convey different information. Some neurons fire in bursts, others fire regularly, others fire irregularly. The pattern of firing, not just the rate, carries information. The action potential travels down the axon like a wave. When one section of the axon depolarizes, it triggers depolarization in the adjacent section, which triggers the next section, and so on. In unmyelinated axons, this wave travels at about 1 meter per second, but many axons are wrapped in myelin, a fatty insulation produced by glial cells. Myelin acts like the insulation on an electrical wire, preventing charge from leaking out. Myelin doesn't cover the entire axon. There are gaps called nodes of Ranvier every millimeter or so. The action potential jumps from node to node, a process called saltatory conduction, which dramatically increases speed. Myelinated axons can conduct action potentials at over 100 meters per second, allowing rapid communication across the nervous system. This speed is crucial for coordinated behavior. When you touch something hot and pull your hand back, signals travel from your finger to your spinal cord and back to muscles in your arm in less than 1/10 of a second. The diversity of ion channels in neurons provides additional complexity. Beyond voltage-gated sodium and potassium channels, there are calcium channels, chloride channels, and many subtypes of each, with different voltage sensitivities, kinetics, and distributions. Different combinations of ion channels give neurons different electrical properties, allowing some neurons to fire rapidly and briefly while others sustain firing, some to oscillate rhythmically while others fire irregularly. This diversity of electrical properties, combined with diversity of connections, creates the rich repertoire of neural dynamics underlying brain function. Synapses are the junctions where neurons communicate. When an action potential reaches the axon terminal, it triggers the release of neurotransmitters, chemical messengers stored in tiny vesicles. Voltage-gated calcium channels open when the action potential arrives, and calcium ions rush into the terminal. Calcium triggers vesicles to fuse with the cell membrane and dump their contents, thousands of neurotransmitter molecules, into the synaptic cleft, the tiny gap between neurons, typically only 20-40 nanometers wide. Neurotransmitters diffuse across this gap in less than a millisecond and bind to receptors on the receiving neuron's membrane. There are two main types of receptors. Ionotropic receptors are ion channels that open when neurotransmitter binds, directly allowing ions to flow across the membrane. This produces fast effects within milliseconds. Metabotropic receptors trigger intracellular signaling cascades when neurotransmitter binds, leading to slower, longer-lasting, and more complex effects. Whether a synapse excites or inhibits the receiving neuron depends on the neurotransmitter and receptor type. Excitatory synapses make the receiving neuron more likely to fire an action potential. Inhibitory synapses make it less likely. A single neuron receives inputs from thousands of other neurons, some excitatory and some inhibitory. At any moment, the neuron integrates all these inputs. If excitation sufficiently exceeds inhibition, the neuron fires. This integration happens continuously, with the neuron's membrane potential fluctuating as inputs arrive. The neuron acts as a coincidence detector, firing when enough excitatory inputs arrive close together in time, and as a pattern recognizer, responding to particular combinations of inputs. Neurotransmitters don't remain in the synaptic cleft indefinitely. They're cleared through several mechanisms. reuptake transporters on the sending neurons, membranes suck neurotransmitters back into the cell for reuse. Enzymes in the synaptic cleft break down neurotransmitters, and diffusion carries neurotransmitters away from the synapse. This rapid clearance ensures each signal is brief and distinct, preventing the nervous system from becoming flooded with neurotransmitter. Many drugs affect synaptic transmission. Cocaine blocks dopamine reuptake transporters, causing dopamine to remain in synapses longer, amplifying its effects. SSRIs, selective serotonin reuptake inhibitors, used to treat depression, block serotonin reuptake transporters. Benzodiazepines, used for anxiety, enhance the effects of GABA, the brain's main inhibitory neurotransmitter. Many toxins interfere with synaptic transmission. Botulinum toxin prevents acetylcholine release, causing paralysis. Nerve gases block the enzyme that breaks down acetylcholine, causing overstimulation of muscles and potentially lethal effects. Synapses aren't static, they change strength with use, a property called synaptic plasticity. When a synapse is repeatedly activated, it can become stronger, requiring less input to produce the same effect in the receiving neuron. This is called long-term potentiation, or LTP. Conversely, synapses that are rarely used can weaken, a process called long-term depression, or LTD. The phrase "neurons that fire together wire together," proposed by Donald Hebb, captures this principle. Synaptic plasticity is thought to be the cellular basis of learning and memory. When you learn something new, specific patterns of synapses strengthen, physically encoding the memory in the brain's structure. This means memory isn't stored in any single location, but is distributed across the patterns of connectivity throughout the brain. The mechanisms of synaptic plasticity are complex and diverse. LTP at many synapses requires activation of NMDA receptors, a type of glutamate receptor with unusual properties. NMDA receptors allow calcium to enter the cell only when two conditions are met simultaneously. Glutamate is bound to the receptor, and the postsynaptic membrane is depolarized. This coincidence detection makes NMDA receptors ideal for Hebbian learning. They strengthen synapses when pre- and postsynaptic neurons are active together. The calcium entering through NMDA receptors triggers molecular cascades that strengthen the synapse, including insertion of more AMPA receptors, another type of glutamate receptor, into the membrane, enlargement of dendritic spines, and synthesis of new proteins. These changes can last hours, days, or even permanently, providing a mechanism for long-term memory storage. Synaptic plasticity occurs at multiple timescales. Short-term plasticity, occurring over milliseconds to minutes, involves changes in neurotransmitter release probability and temporary depletion or enhancement of available vesicles. This provides neurons with working memory, the ability to hold information briefly for immediate processing. Long-term plasticity, requiring protein synthesis and structural changes, provides the basis for more permanent memory storage. The diversity of plasticity mechanisms allows the brain to adapt flexibly to experience across different timescales, from moment-to-moment adjustments to lifelong learning. The brain is divided into several major regions, each with specialized functions. Starting from the bottom and working up, the brainstem is the oldest part of the brain evolutionarily and controls basic life-sustaining functions. The medulla oblongata at the very base of the brainstem regulates heartbeat, breathing, blood pressure, and reflexes like swallowing and vomiting. Damage to the medulla is typically fatal, because it disrupts these essential functions. The medulla contains respiratory centers that monitor carbon dioxide levels in blood and adjust breathing rate accordingly. It contains cardiovascular centers that regulate blood pressure by controlling heart rate and blood vessel constriction. It contains reflex centers for coughing, sneezing, swallowing, and vomiting, protective reflexes that clear airways or expel harmful substances. Above the medulla is the pons, which relays signals between the cerebrum and cerebellum and also contributes to regulating breathing and sleep. The pons contains nuclei involved in REM sleep generation, and damage to the pons can disrupt sleep-wake cycles. The name pons means bridge in Latin, reflecting its role in connecting different brain regions. The midbrain at the top of the brainstem is involved in vision, hearing, motor control, and arousal. The superior colliculus controls eye movements and visual attention, helping orient toward important stimuli. The inferior colliculus processes auditory information and helps localize sounds. The substantia nigra, part of the midbrain, produces dopamine and is crucial for movement. Degeneration of substantia nigra neurons causes Parkinson's disease. The ventral tegmental area, adjacent to the substantia nigra, also produces dopamine and is central to reward and motivation. The periaqueductal gray, surrounding the cerebral aqueduct that carries cerebrospinal fluid through the midbrain, is involved in pain modulation. Stimulation of this area can produce powerful analgesia, apparently by activating descending pain inhibitory pathways. Attached to the back of the brainstem is the cerebellum, Latin for little brain. The cerebellum is remarkably compact. Though it constitutes only about 10% of the brain's total volume, it contains more than half of all the brain's neurons, about 50 billion neurons in a structure the size of a fist. These neurons are organized in a highly regular, repeating pattern, unlike the more variable organization in other brain regions. The cerebellum is crucial for motor control, coordinating movements to be smooth, precise, and properly timed. It doesn't initiate movements, that's the motor cortex's job, but it refines them, adjusting ongoing movements based on sensory feedback and predicting the sensory consequences of movements. When you reach for a cup, your motor cortex initiates the movement, but your cerebellum ensures your hand travels smoothly to the right location, adjusts for the cup's weight as you lift it, and prevents your hand from overshooting or moving jerkily. Damage to the cerebellum doesn't cause paralysis, but causes ataxia. Movements become jerky, clumsy, and poorly coordinated. Walking becomes unsteady, with a wide-based gait. Reaching movements overshoot or undershoot targets. Speech becomes slurred because the precise coordination of mouth and throat muscles is impaired. Eye movements become jerky. The cerebellum also contributes to cognitive functions, particularly those involving precise timing and sequencing, and may play a role in language, attention, and emotional processing. Recent research suggests the cerebellum is involved in predicting not just sensory consequences of movements, but also cognitive and emotional consequences of actions, contributing to learning and decision-making more broadly. The cerebellum learns from errors. When movements don't produce expected outcomes, cerebellar circuits adjust to improve future performance. This error-based learning allows acquisition of motor skills through practice and adaptation to changing conditions like injury or using tools. Above the brainstem is the diencephalon, which includes the thalamus and hypothalamus. The thalamus is a collection of nuclei that serves as a relay station. Almost all sensory information, except smell, passes through the thalamus on its way to the cerebral cortex. Different thalamic nuclei relay different types of information. Visual information goes through the lateral geniculate nucleus, auditory through the medial geniculate nucleus, somatosensory through the ventral posterior nucleus, and so on. The thalamus doesn't just passively relay information, but actively filters and modulates it, influenced by attention, arousal, and other factors. Attention can enhance thalamic transmission of attended information while suppressing unattended information, helping focus cortical processing on relevant inputs. The thalamus also participates in generating rhythmic brain activity, particularly the oscillations characteristic of sleep and certain pathological states like absence epilepsy. The hypothalamus, despite being only about the size of an almond, regulates fundamental drives and homeostasis. It controls hunger, thirst, body temperature, sleep-wake cycles, and the autonomic nervous system. It monitors body temperature, blood glucose, blood osmolarity, and hormones, adjusting behavior and physiology to maintain homeostasis. When blood glucose drops, the hypothalamus triggers hunger and activates hormonal responses that raise glucose. When body temperature rises, the hypothalamus triggers sweating and vasodilation to promote heat loss. The hypothalamus controls the pituitary gland, the master gland that produces hormones regulating growth, metabolism, reproduction, and stress responses. The hypothalamus produces releasing hormones that control pituitary hormone secretion, linking the nervous system to the endocrine system. Damage to specific parts of the hypothalamus can cause overeating or loss of appetite, disrupted sleep, inability to regulate body temperature, or hormonal imbalances. The hypothalamus is organized into nuclei with distinct functions. The suprachiasmatic nucleus regulates circadian rhythms. The paraventricular nucleus produces hormones regulating water balance and stress. The ventromedial nucleus influences satiety. The lateral hypothalamus influences hunger, and the preoptic area regulates temperature and sleep. The limbic system is a group of structures involved in emotion, motivation, and memory. Key limbic structures include the amygdala, involved in processing emotions, particularly fear; the hippocampus, essential for forming new memories; the cingulate cortex, involved in emotion regulation and decision-making; and parts of the basal ganglia, involved in reward and motivation. The limbic system is densely interconnected with the hypothalamus and prefrontal cortex, creating integrated circuits that link emotion, motivation, memory, and decision-making. The concept of the limbic system as a distinct emotional brain has been criticized because many so-called limbic structures have non-emotional functions, and emotion involves brain regions beyond the traditional limbic system. Nevertheless, these structures work together in processing emotionally significant information and linking it to memory, motivation, and physiological responses. The cerebral cortex is the wrinkled outer layer of the cerebrum responsible for higher cognitive functions. The cortex is only about 2-4 millimeters thick, but has a surface area of about 2,500 square centimeters, roughly the size of a large pizza. How does such a large surface fit inside the skull? Through extensive folding. The cortex is folded into ridges called gyri, singular, gyrus, and grooves called sulci, singular, sulcus. Particularly deep sulci are called fissures. These folds increase surface area without requiring a larger skull. The cortex is divided into two hemispheres, left and right, which are separated by the deep longitudinal fissure but connected by the corpus callosum, a thick bundle of about 200 million nerve fibers that allows the hemispheres to communicate. Each hemisphere is divided into 4 lobes, named after the skull bones that cover them. The frontal lobe, the largest lobe, is involved in motor control, planning, decision-making, personality, and language production. The very front portion, the prefrontal cortex, is particularly important for executive functions, planning, impulse control, working memory, and abstract reasoning. The prefrontal cortex is one of the last brain regions to fully mature, not reaching full development until the mid-20s, which may partly explain why adolescents and young adults are more prone to impulsive and risky behavior than older adults. The motor cortex, located in the posterior part of the frontal lobe along the central sulcus, controls voluntary movements. It's organized as a map of the body, with specific areas controlling specific body parts. Like the somatosensory map, this motor map is distorted, body parts requiring fine motor control, like hands and lips, occupy disproportionately large cortical areas. Stimulating specific points in motor cortex with electrodes elicits movements of corresponding body parts, a technique used during brain surgery to map motor function and avoid damaging critical areas. Behind the frontal lobe, separated by the central sulcus, is the parietal lobe, which processes sensory information about touch, temperature, pain, and body position. The front part of the parietal lobe contains the primary somatosensory cortex, which receives touch information from the body. This region is organized as a map of the body, with specific areas processing input from specific body parts. Interestingly, the map is distorted. Areas with high tactile sensitivity, like the hands, lips, and tongue, occupy disproportionately large regions of cortex relative to their body size. This distorted map is called a sensory homunculus, and if you drew a person with body parts sized according to their cortical representation, they'd have enormous hands and lips and a tiny torso and legs. The posterior parietal cortex integrates sensory information from different modalities and is involved in spatial awareness, attention, and visually guided action. Damage here can cause neglect syndrome, where patients fail to attend to one side of space, ignoring objects, people, or even their own body parts on the neglected side. The temporal lobe, below the frontal and parietal lobes, is involved in hearing, language comprehension, memory, and recognizing faces and objects. The primary auditory cortex, which receives sound information, is located on the upper surface of the temporal lobe, in a region called Heschl's gyrus. Auditory cortex is organized tonotopically. Different regions respond preferentially to different sound frequencies, creating a map of pitch along the cortical surface. Deeper within the temporal lobe are structures crucial for memory, including the hippocampus and surrounding cortical regions. The inferior temporal cortex is involved in visual object recognition. Neurons here respond selectively to complex visual patterns like faces, bodies, and familiar objects. Damage to specific parts of the temporal cortex can cause difficulty recognizing faces, prosopagnosia, understanding language, Wernicke's aphasia, or forming new memories, amnesia. The occipital lobe at the very back of the brain is dedicated to vision. The primary visual cortex, which receives visual information from the eyes, is located here, surrounding the calcarine sulcus on the medial surface of the occipital lobe. The primary visual cortex is organized retinotopically. Neighboring areas of cortex represent neighboring areas of the visual field, creating a distorted map of visual space. The central region of vision, the fovea, where acuity is highest, occupies a disproportionately large area of visual cortex. The primary visual cortex contains neurons that respond to specific features like edges, orientations, and directions of motion. Visual information is then sent to surrounding areas for more complex processing. The visual system is organized into multiple pathways. The ventral stream, projecting toward the temporal lobe, processes what an object is, analyzing form, color, and identity. The dorsal stream, projecting toward the parietal lobe, processes where an object is and how it's moving, analyzing location, motion, and spatial relationships. These streams work together to create coherent visual perception, though damage to one stream can selectively impair certain aspects of vision. Ventral stream damage might impair object recognition while preserving spatial vision, while dorsal stream damage might impair reaching for objects while preserving recognition. The cortex isn't uniformly organized, but has distinct types of cortex with different structures and functions. Primary sensory and motor areas have characteristic laminar structure, 6 layers of neurons with different patterns of connections. Sensory cortex has a thick layer 4, which receives thalamic inputs, while motor cortex has a thick layer 5, which sends outputs to the spinal cord and brain stem. Association cortex, which integrates information from multiple sensory modalities and supports higher cognitive functions, has more variable structure with extensive interconnections between layers. The cortex also shows columnar organization. Neurons form vertical columns through the cortical layers, with neurons in a column sharing similar response properties and forming local circuits that process related information. This organization was discovered by Hubel and Wiesel in visual cortex, where they found neurons organized into orientation columns, with each column responding to edges of a particular orientation, and ocular dominance columns, with each column responding preferentially to input from one eye. The 2 hemispheres of the brain are specialized for different functions. This lateralization of function means that certain abilities are primarily handled by one hemisphere or the other, though both hemispheres contribute to most tasks to some degree. The most obvious lateralization is for motor control and sensation. The left hemisphere controls the right side of the body and receives sensory input from the right side, while the right hemisphere controls the left side of the body and receives input from the left side. This crossover occurs because nerve fibers cross to the opposite side as they travel through the brainstem. The crossing is nearly complete for motor and sensory systems, though some ipsilateral, same-side, pathways exist, particularly for trunk and proximal limb control. Language is also lateralized. For about 95% of right-handed people and about 70% of left-handed people, language is primarily processed in the left hemisphere. Broca's area in the left frontal lobe is involved in speech production and grammatical processing. Damage causes Broca's aphasia, where people can understand language but have difficulty speaking, producing slow, effortful, telegraphic speech with grammar errors. Wernicke's area in the left temporal lobe is involved in language comprehension. Damage causes Wernicke's aphasia, where people produce fluent but meaningless speech and have difficulty understanding language. The arcuate fasciculus, a bundle of fibers connecting Wernicke's and Broca's areas, is also crucial. Damage here causes conduction aphasia, where comprehension and production are relatively preserved, but repetition is impaired. Beyond these classical language areas, other left hemisphere regions contribute to language, including the angular gyrus, involved in reading, and parts of the basal ganglia and thalamus. Other left hemisphere specializations include sequential processing, analytical thinking, processing details, and mathematical reasoning. The left hemisphere tends to process information sequentially and analytically, breaking down complex information into components. The right hemisphere has its own specializations. It's more involved in spatial processing, face recognition, emotional processing, particularly negative emotions, understanding metaphors and jokes, prosody, the melody and emotional tone of speech, and processing global patterns rather than details. The right hemisphere tends to process information holistically, integrating components into wholes. Right hemisphere damage can make speech sound flat and monotone and make it difficult to understand others' emotional tone, to appreciate humor, or to navigate spatial environments. Right hemisphere stroke can cause spatial neglect, where patients ignore the left side of space. Split brain patients provide dramatic evidence of hemispheric specialization. These are patients who had their corpus callosum surgically severed to control severe epilepsy. After the surgery, the two hemispheres can no longer communicate directly. Researchers can present information to just one hemisphere, for example, flashing a word to the left visual field, which goes to the right hemisphere. Split-brain patients can't verbally report seeing the word because the left hemisphere, which controls speech, didn't see it. But if asked to point with their left hand, controlled by the right hemisphere, they can point to the correct object. This shows that each hemisphere has its own perceptions and knowledge, normally unified through communication across the corpus callosum, but separable when that connection is cut. Split brain studies reveal that the two hemispheres can have different preferences, knowledge, and even conflicting intentions. One patient reported that sometimes his left hand would select different clothes than his right hand intended, or would reach for objects that his speaking self didn't want. However, it's important not to overstate hemispheric differences. Popular psychology often exaggerates lateralization, claiming people are left-brained, logical, analytical, or right-brained, creative, intuitive. Brain imaging studies show that people don't preferentially use one hemisphere. Virtually all tasks involve both hemispheres, and individual differences in thinking style don't correspond to differences in hemispheric dominance. The brain works as an integrated whole, with the two hemispheres constantly communicating and collaborating. Hemispheric specialization is a matter of degree, not absolute division, and the advantages of specialization must be balanced against the need for integration. Why the brain evolved hemispheric specialization is debated. Proposed advantages include allowing simultaneous processing of different types of information, increasing processing capacity, and enabling complementary processing strategies that together provide more robust cognition than either strategy alone. Neurotransmitters are the chemical messengers that allow neurons to communicate. There are over 100 known neurotransmitters, but a few are particularly important and well studied. Glutamate is the brain's primary excitatory neurotransmitter. Most excitatory synapses in the brain use glutamate. It's crucial for learning and memory. When you learn something, specific patterns of glutamate transmission strengthen, encoding the memory. Glutamate acts on several types of receptors, including AMPA, NMDA, and kainate receptors, each with different properties and roles. However, too much glutamate is toxic. Excessive glutamate release, which can occur during strokes when neurons are deprived of oxygen, causes overactivation of receptors, allowing too much calcium to enter neurons, triggering cell death. This excitotoxicity kills neurons and expands stroke damage beyond the initial area deprived of blood. Excitotoxicity also contributes to neuronal death in neurodegenerative diseases like Alzheimer's and Huntington's disease. GABA, gamma-aminobutyric acid, is the brain's primary inhibitory neurotransmitter. GABA reduces neuronal activity, preventing excessive excitation. The balance between glutamate and GABA is crucial. Too much excitation relative to inhibition can cause seizures, where large populations of neurons fire synchronously and uncontrollably. Many anti-epileptic drugs work by enhancing GABA transmission or reducing glutamate transmission. GABA acts on two main receptor types: GABA receptors, which are ionotropic and produce fast inhibition, and GABAB receptors, which are metabotropic and produce slower, longer-lasting inhibition. GABA is also important for anxiety. Benzodiazepines like Valium and Xanax enhance GABA's effects at GABA receptors, producing calming and anxiety-reducing effects. Alcohol also enhances GABA receptor function, contributing to its sedative effects. Dopamine is involved in reward, motivation, movement, and attention. Dopamine is produced by neurons in the midbrain and released in several target areas, including the striatum, involved in movement, the nucleus accumbens, involved in reward, and the prefrontal cortex, involved in executive functions. The dopamine system is activated by rewards and by cues predicting rewards. When you eat something delicious, use drugs, or achieve a goal, dopamine is released, creating a sense of pleasure and reinforcing the behavior that led to the reward. The dopamine system doesn't just respond to rewards, but learns to predict them. Once a reward becomes predictable, dopamine neurons fire not when the reward is received, but when the cue predicting it appears, creating motivation to pursue rewards. Drugs of abuse, from cocaine to heroin to methamphetamine, all increase dopamine signaling, hijacking the brain's natural reward system. Cocaine blocks dopamine reuptake, while amphetamines both block reuptake and increase dopamine release. Opioids indirectly increase dopamine by inhibiting GABA neurons that normally suppress dopamine neurons. Over time, drug use can dysregulate the dopamine system, making natural rewards less satisfying and increasing craving for the drug. The brain adapts to chronic drug use by reducing dopamine receptor numbers or sensitivity, creating tolerance and withdrawal. Dopamine is also crucial for movement. Parkinson's disease is caused by the death of dopamine-producing neurons in the substantia nigra, a midbrain structure. Loss of dopamine causes the motor symptoms of Parkinson's: tremor, rigidity, slow movement, and difficulty initiating movement. Treatment with L-Dopa, a dopamine precursor that can cross into the brain and be converted to dopamine, can alleviate symptoms, though it doesn't stop the underlying neuronal death and can lose effectiveness over time as more neurons die. Serotonin regulates mood, sleep, appetite, and impulse control. Most serotonin in the body is actually in the gut, not the brain, where it regulates digestion, but brain serotonin has profound effects on behavior and mental health. Serotonin is produced by neurons in the raphe nuclei of the brainstem and released widely throughout the brain. Low serotonin has been implicated in depression, though the relationship is more complex than the simplistic chemical imbalance model suggests. SSRIs, selective serotonin reuptake inhibitors, like Prozac and Zoloft, block the reuptake of serotonin from synapses, increasing serotonin availability. SSRIs are effective for depression, anxiety disorders, and OCD, though they don't work for everyone, can take weeks to produce benefits and often cause side effects. The mechanism by which SSRIs improve mood is still debated. Increasing serotonin is just the immediate effect, but mood improvement requires weeks, suggesting downstream changes in neural circuits and plasticity are necessary. Serotonin also regulates sleep and aggression. Low serotonin is associated with insomnia and impulsive aggression and violence. Serotonin acts on numerous receptor subtypes, at least 14 distinct types, each with different distributions and functions, allowing serotonin to have diverse effects on different neural circuits. Some hallucinogenic drugs, including LSD and psilocybin, work by activating specific serotonin receptors, particularly the 5-HT2A receptor. Norepinephrine, also called noradrenaline, is involved in arousal, attention, and the stress response. Norepinephrine is released throughout the brain during alertness and in response to novelty or danger. It enhances attention and memory formation, which is why emotionally arousing events are remembered better than neutral events. The norepinephrine released during arousal strengthens memory consolidation. Norepinephrine is also released in the body as part of the fight-or-flight response, increasing heart rate, blood pressure, and blood flow to muscles. Some antidepressants target norepinephrine as well as serotonin, SNRIs, serotonin norepinephrine reuptake inhibitors, block reuptake of both neurotransmitters. Acetylcholine is involved in attention, learning, memory, and muscle contraction. In the brain, acetylcholine is released by neurons in the basal forebrain and brainstem that project widely throughout the cortex. Acetylcholine levels are highest during waking and REM sleep, when attention and learning are possible, and lowest during deep sleep. Acetylcholine enhances cortical responses to sensory stimuli and facilitates synaptic plasticity, promoting learning. Acetylcholine is also the neurotransmitter at the neuromuscular junction, where motor neurons communicate with muscles. Blocking acetylcholine causes paralysis, which is how botulinum toxin and many other toxins work. Alzheimer's disease involves degeneration of acetylcholine-producing neurons, contributing to memory and cognitive deficits. Some Alzheimer's treatments work by inhibiting acetylcholinesterase, the enzyme that breaks down acetylcholine, increasing its availability, though these drugs provide only modest benefits and don't stop disease progression. Beyond these classical neurotransmitters, many other molecules function as neurotransmitters or neuromodulators. Endorphins and enkephalins are endogenous opioids that modulate pain and produce pleasure. Anandamide and 2-AG are endocannabinoids that modulate pain, appetite, and mood, and are the targets of THC, the psychoactive component of cannabis. Nitric oxide is an unusual neurotransmitter, a gas that diffuses through membranes rather than being released from vesicles, involved in blood vessel regulation and possibly in synaptic plasticity. Neuropeptides like substance P, oxytocin, and vasopressin modulate pain, social bonding, and fluid regulation, respectively. The diversity of neurotransmitter systems allows for complex modulation of neural activity, with different systems activated in different contexts to adjust brain state and behavior appropriately. The basal ganglia are a group of structures deep in the brain involved in movement and learning. The basal ganglia include the striatum, divided into the caudate nucleus and putamen, the globus pallidus, the substantia nigra, and the subthalamic nucleus. These structures form loops with the cortex. The cortex sends signals to the striatum, which processes them through the internal circuitry of the basal ganglia, and then sends signals to the thalamus, which projects back to the cortex, completing the loop. These corticobasal ganglia-thalamocortical loops are involved in selecting and executing actions. The basal ganglia receive information about the current situation and internal goals from the cortex, evaluate possible actions, select the most appropriate action, and facilitate its execution while inhibiting competing actions. This process normally happens unconsciously and rapidly. When you reach for a cup of coffee, the basal ganglia help select the appropriate motor program. The sequence of muscle activations needed to move your arm, open your hand, grasp the cup with the right grip force, and lift it, while simultaneously inhibiting other possible actions like scratching your head or checking your phone. Damage to the basal ganglia disrupts movement in characteristic ways. Parkinson's disease, caused by loss of dopamine neurons in the substantia nigra, produces slow movement bradykinesia, difficulty initiating movement, akinesia, rigidity, and tremor. Patients with Parkinson's often describe feeling like they're moving through molasses. The motor programs exist, but they can't be executed smoothly. The basal ganglia dysfunction in Parkinson's doesn't prevent movement entirely, but makes it slow, effortful, and poorly initiated. Interestingly, Parkinson's patients sometimes retain the ability to make automatic, habitual movements better than voluntary movements, and external cues like lines on the floor can help initiate walking when internal initiation fails, suggesting the basal ganglia are particularly important for self-initiated, goal-directed actions. Huntington's disease, caused by degeneration in the striatum, produces involuntary, jerky movements called chorea, along with cognitive and psychiatric problems. The movements in Huntington's reflect loss of the basal ganglia's normal function of inhibiting unwanted movements. Without this inhibition, motor programs are released inappropriately, causing the characteristic dance-like movements. Huntington's is caused by an expanded CAG repeat in the Huntingtin gene, which produces a mutant protein that gradually kills striatal neurons. The disease is inherited in an autosomal dominant pattern, and onset is typically in midlife, though age of onset and severity correlate with repeat length. There's currently no cure, though research into gene therapies and other approaches is ongoing. The basal ganglia aren't only involved in motor control, they also contribute to cognitive and emotional functions. Different parts of the striatum process different types of information. The dorsal striatum receives input from motor and sensory cortex and is primarily involved in movement and habit learning. The ventral striatum, particularly the nucleus accumbens, receives input from limbic and prefrontal areas and is involved in motivation, reward, and emotion. Damage to the ventral striatum can cause apathy and loss of motivation. The basal ganglia play a crucial role in habit formation and procedural learning skills like riding a bike, playing a musical instrument, or driving a car. These skills, once learned, become automatic and don't require conscious attention. This learning involves the basal ganglia's ability to reinforce frequently performed action sequences, gradually making them habitual. Habits are powerful. They're executed automatically in response to cues without conscious decision-making, which is efficient but can be problematic when trying to change unwanted habits. The reinforcement learning process in the basal ganglia depends on dopamine. When an action leads to a better-than-expected outcome, dopamine is released, strengthening the connections that led to that action. When an outcome is worse than expected, dopamine dips below baseline, weakening those connections. This creates a learning signal that helps the brain discover which actions lead to rewards. This process, formalized in computational models as temporal difference learning, is remarkably similar to reinforcement learning algorithms used in artificial intelligence. Addiction involves dysregulation of this system. Drugs produce massive dopamine release, creating a powerful learning signal that reinforces drug-seeking behavior, making it increasingly automatic and compulsive. Over time, drug-associated cues trigger dopamine release and craving even without drug consumption, and the brain adapts to chronic drug exposure by reducing baseline dopamine, creating a state where natural rewards no longer satisfy, and the addicted person needs drugs just to feel normal. The basal ganglia are also implicated in various psychiatric disorders. Obsessive-compulsive disorder involves hyperactivity in corticobasal ganglia loops, possibly related to difficulty inhibiting unwanted thoughts and actions. Tourette syndrome, characterized by motor and vocal tics, involves basal ganglia dysfunction affecting motor inhibition. Schizophrenia involves dopamine dysregulation in basal ganglia circuits, and antipsychotic medications work primarily by blocking dopamine receptors in these circuits. The basal ganglia's involvement in such diverse functions and disorders reflects their central role in action selection, motivation, and learning. The hippocampus is essential for forming new long-term memories. Located in the medial temporal lobe, the hippocampus is a curved structure about the size and shape of a seahorse. Hippocampus is Greek for seahorse. You actually have two hippocampi, one in each hemisphere. The hippocampus is part of the limbic system and is crucial for episodic memory, memory for events and experiences, and spatial memory, memory for locations and navigation. The importance of the hippocampus was dramatically demonstrated by patient HM, one of the most famous cases in neuroscience. HM, whose full name was Henry Molaison, revealed after his death in 2008, suffered from severe epilepsy that couldn't be controlled with medication. In 1953, in a last resort treatment, surgeons removed most of his medial temporal lobes bilaterally, including both hippocampi and surrounding cortical regions. The surgery successfully reduced his seizures, but it had a devastating side effect. HM could no longer form new long-term memories. He could hold information in working memory. If you told him a number, he could repeat it back immediately, and if he focused on it without distraction, he could maintain it for several minutes through rehearsal. But if his attention was distracted even briefly, the information was lost. He couldn't remember meeting someone 5 minutes earlier. He couldn't learn where he lived after the surgery or remember what he'd eaten for breakfast. Every experience was fresh because he couldn't form memories of it. He lived in an eternal present, unable to remember the recent past or anticipate the future based on recent experience. Remarkably, HM's memories from before the surgery were largely intact. He could remember his childhood, his early adulthood, and events from years before the surgery. This showed that the hippocampus is necessary for forming new long-term memories, a process called encoding, but not for storing them long-term or retrieving old memories. Over time, memories apparently become independent of the hippocampus through a process called consolidation, where they're gradually transferred to other brain regions, primarily the cortex, for long-term storage. This consolidation process takes time. Memories from the years immediately before HM's surgery were impaired, retrograde amnesia, while more remote memories were spared, suggesting consolidation takes months to years. HM also retained the ability to learn new motor skills. He could learn to trace a shape while looking at his hand in a mirror, a difficult task requiring practice. His performance improved over days of practice, demonstrating learning, but each day he had no memory of having done the task before. This demonstrated that different types of memory, explicit memory, conscious memory for facts and events, and implicit memory, unconscious memory expressed through performance, depend on different brain systems. The hippocampus is required for explicit memory, but not implicit memory. This dissociation has been confirmed in many other patients and in experimental animals. The hippocampus is particularly important for spatial memory and navigation. Taxi drivers in London, who must memorize the complex layout of thousands of streets, a process called the knowledge that takes years of study, have larger posterior hippocampi than controls, and the size correlates with years of experience. Studies of rodents show that the hippocampus contains place cells that fire when the animal is in a specific location, creating a neural map of space. These place cells, discovered by John O'Keefe, who shared the 2014 Nobel Prize in Physiology or Medicine, along with the discovery of grid cells in the nearby entorhinal cortex by May Britt and Edvard Moser, who shared the same Nobel Prize, have revolutionized understanding of how the brain represents space. Different place cells fire in different locations, creating a population code that represents the animal's position. The firing of place cells is influenced not just by location, but by direction of movement, goals, and context, suggesting the hippocampus integrates spatial information with other information to create rich cognitive maps. The hippocampus is one of the few brain regions where new neurons continue to be generated throughout life, a process called adult neurogenesis. New neurons are born in the dentate gyrus, a subregion of the hippocampus, migrate into the granule cell layer, and integrate into existing circuits. Exercise, learning, and enriched environments promote neurogenesis, while stress inhibits it. The function of adult neurogenesis is still being studied, but it may contribute to the hippocampus's capacity for learning new information and distinguishing similar memories, pattern separation. The rate of neurogenesis declines with age, which may contribute to age-related memory decline. The hippocampus is also vulnerable to damage. It's one of the first brain regions affected by Alzheimer's disease, which is why memory problems are an early symptom. In Alzheimer's, abnormal protein aggregates, amyloid plaques, and tau tangles accumulate in the hippocampus and other brain regions, causing neuronal dysfunction and death. The hippocampus is also highly sensitive to oxygen deprivation, so stroke or cardiac arrest can damage it, causing memory impairments even if the person survives. Prolonged stress and elevated cortisol levels can shrink the hippocampus, and hippocampal volume is reduced in depression and PTSD, which may contribute to the memory problems associated with these conditions. However, the hippocampus retains some plasticity, and hippocampal volume can increase with interventions like exercise, suggesting some reversibility of stress-related damage. The amygdala is central to processing emotions, particularly fear. The amygdala is an almond-shaped structure. Amygdala is Greek for almond, located in the medial temporal lobe, just in front of the hippocampus. Like the hippocampus, you have two amygdalae, one in each hemisphere. The amygdala is actually a complex of several nuclei with different functions and connections. The amygdala receives input from all sensory systems and has extensive connections to many other brain regions, including the hippocampus, prefrontal cortex, hypothalamus, and brainstem. This connectivity allows the amygdala to monitor the environment for emotionally significant stimuli and orchestrate appropriate responses. The amygdala is best known for its role in fear. When you encounter a threat, or even a stimulus that resembles a threat, the amygdala activates within milliseconds, triggering a cascade of responses. The amygdala sends signals to the hypothalamus, activating the sympathetic nervous system and stress hormone release, the fight-or-flight response. It sends signals to the brainstem, modulating reflexes, facial expressions, and pain sensitivity. It influences the hippocampus, enhancing memory formation for the threatening event. This is why emotionally arousing events are remembered vividly, and it communicates with the cortex, bringing the threat into conscious awareness. Importantly, there are two pathways from sensory systems to the amygdala, a fast, direct pathway that bypasses the cortex, and a slower pathway that goes through the cortex. The fast pathway, called the low road, allows rapid responses to threats before you've consciously perceived them. This is why you might jump at a sudden sound or flinch at a looming object before you've consciously identified what it was. The conscious processing comes through the slower cortical pathway, the high road, allowing you to evaluate whether the threat is real and modulate your response accordingly. If you jump at a sound and then realize it was just a door closing, the cortical pathway has informed the amygdala that there's no real threat, allowing the fear response to subside. This two-pathway system provides a balance between speed, responding quickly to potential threats, and accuracy, not responding to false alarms. Fear conditioning is a well-studied form of learning that depends on the amygdala. If a neutral stimulus, like a tone, is paired with an unpleasant stimulus, like a mild shock, the neutral stimulus becomes a conditioned fear stimulus, triggering fear responses even without the shock. This learning occurs in the amygdala. Lesioning the amygdala prevents fear conditioning, and neurons in the amygdala, particularly the lateral nucleus, change their firing patterns during fear learning, strengthening responses to the conditioned stimulus. This process is adaptive. It allows you to learn what's dangerous and respond quickly to potential threats. However, sometimes fear conditioning becomes maladaptive, contributing to anxiety disorders and phobias. A person who experiences a panic attack in an elevator might develop a conditioned fear of elevators, or even of small spaces in general, leading to avoidance behavior that interferes with daily life. PTSD involves dysregulated fear processing, with the amygdala becoming hyperresponsive to trauma-related cues and failing to extinguish fear responses even in safe contexts. Exposure therapy, a treatment for anxiety disorders and phobias, works by repeatedly exposing the person to the feared stimulus in a safe context, allowing new learning that the stimulus is not dangerous. This process, called extinction, doesn't erase the original fear memory, but creates a new, competing memory that inhibits the fear response. Extinction learning involves the prefrontal cortex, particularly the ventromedial prefrontal cortex, learning that the stimulus is safe and sending inhibitory signals to reduce the amygdala's fear response. Importantly, extinction is context-dependent. A fear response extinguished in one setting can return in a different setting, renewal, or after time has passed, spontaneous recovery, or after a stressful event, reinstatement, suggesting the original fear memory remains and can be reactivated. This is why anxiety treatments often include practicing coping strategies in multiple contexts and building skills to manage anxiety when it returns. While the amygdala is often associated with fear, it's involved in processing other emotions too, including positive emotions. The amygdala responds to pleasant stimuli like attractive faces, pleasant smells, and rewards, not just threats. More generally, the amygdala seems to detect emotional salience, anything important or intense, positive or negative, and modulate attention, memory, and behavior accordingly. The amygdala evaluates the biological significance of stimuli, determining whether they're important enough to warrant further processing and response. This broader role is consistent with the amygdala's involvement in social cognition. It responds strongly to faces, particularly emotional expressions, and helps interpret others' emotions and intentions. People with amygdala damage often have difficulty recognizing fear and anger in others' faces and may show reduced social awareness, though social functioning isn't completely eliminated. The amygdala is part of a network including the fusiform face area, superior temporal sulcus, and prefrontal cortex that processes social information and guides social behavior. The prefrontal cortex is involved in executive functions, planning, decision-making, and impulse control. The prefrontal cortex is the frontmost part of the frontal lobe, just behind the forehead. It's proportionally larger in humans than in other species, constituting about 30% of the cortex in humans compared to about 17% in chimpanzees and 7% in dogs. This expansion of the prefrontal cortex is thought to underlie humans' advanced cognitive abilities, including abstract reasoning, planning over long time horizons, language, and complex social behavior. The prefrontal cortex can be divided into several regions with somewhat different functions. The dorsolateral prefrontal cortex is involved in working memory, cognitive flexibility, planning, and abstract reasoning. Working memory, the ability to hold and manipulate information temporarily, depends heavily on this region. Neurons in dorsolateral prefrontal cortex maintain sustained activity during working memory tasks, representing information that must be held in mind. Damage to dorsolateral prefrontal cortex impairs the ability to plan complex actions, think abstractly, hold multiple pieces of information in mind simultaneously, or shift flexibly between tasks. Patients may perseverate, continuing to use strategies that no longer work, unable to shift to new approaches. They may have difficulty organizing behavior toward goals, starting tasks but not completing them, or jumping between tasks without finishing any. The ventromedial prefrontal cortex is involved in decision-making, emotion regulation, and evaluating rewards and punishments. This region receives input from the amygdala and other limbic structures, integrating emotional information with cognitive processing to guide decisions. The famous case of Phineas Gage illustrates the ventromedial prefrontal cortex's importance. Gage was a railroad construction foreman who, in 1848, survived an accident where an iron rod was driven completely through his head, entering beneath his left cheekbone and exiting through the top of his skull, damaging his ventromedial prefrontal cortex. Remarkably, Gage survived, retained his memory and intellectual abilities, could speak and move, and recovered physically. But his personality changed dramatically. Friends said he was no longer Gage. He became impulsive, profane, irresponsible, unable to make good decisions or plan for the future, and couldn't maintain employment. Modern analysis of Gage's skull and contemporary accounts suggests the damage was largely to the ventromedial prefrontal cortex, supporting this region's role in personality, social behavior, and decision-making. Modern patients with ventromedial prefrontal damage show similar patterns: preserved intellect and memory, but impaired decision-making, particularly in personal and social domains. They make poor financial decisions, engage in risky behavior, have difficulty maintaining relationships, and seem unable to use emotional information to guide choices. Laboratory tests reveal that they fail to develop anticipatory emotional responses, measured as skin conductance, before making risky choices in gambling tasks, while healthy controls show these responses. suggesting that the ventromedial prefrontal cortex uses emotional signals to mark choices as potentially good or bad, guiding decisions even before conscious deliberation. Without this emotional guidance, decisions become divorced from their likely consequences. The orbitofrontal cortex, on the underside of the frontal lobe, is involved in evaluating rewards and punishments, inhibiting inappropriate behavior, and processing sensory information in the context of reward. Damage here can cause impulsive behavior, poor decision-making, and difficulty learning from mistakes. Patients with orbitofrontal damage may repeatedly make choices that lead to negative consequences, seemingly unable to use feedback to guide behavior. They may engage in socially inappropriate behavior, making offensive comments or violating social norms without apparent awareness of the inappropriateness. The orbitofrontal cortex is thought to represent the value of different options, updating these representations based on outcomes and using them to guide choices toward rewarding and away from punishing outcomes. The prefrontal cortex is heavily interconnected with other brain regions, allowing it to influence virtually all aspects of cognition and behavior. It receives information from sensory and motor cortex, the limbic system, and the basal ganglia. It sends signals back to these same regions, modulating their activity based on goals, plans, and context. This top-down control allows flexible, goal-directed behavior rather than purely stimulus-driven responses. The prefrontal cortex can suppress habitual responses when they're inappropriate, delay gratification for long-term benefits, and adjust behavior based on changing circumstances. These abilities are central to human success and vary considerably across individuals and across the lifespan. The prefrontal cortex doesn't fully mature until the mid-20s. Throughout childhood and adolescence, the prefrontal cortex undergoes synaptic pruning and myelination, gradually becoming more efficient. Gray matter volume in prefrontal cortex actually decreases during adolescence as synapses are pruned, while white matter increases as axons are myelinated. These changes improve the efficiency and speed of prefrontal processing. This protracted development may contribute to adolescents' tendency toward risk-taking, impulsive behavior, and difficulty with long-term planning. The prefrontal cortex is developing while the limbic system is mature and highly reactive, creating an imbalance where emotional and reward-driven impulses can override deliberative control. This isn't a flaw but may have evolutionary advantages. Adolescence is a time for exploration, learning, and establishing independence, and some risk-taking may facilitate these goals. The prefrontal cortex is also one of the brain regions most sensitive to stress. Acute stress can actually improve prefrontal function, enhancing focus and performance. But chronic stress impairs prefrontal function, reducing working memory, decision-making, and impulse control, while simultaneously enhancing amygdala function and habit-based responding. This shift makes sense from a survival perspective. In immediate danger, you don't need careful deliberation. You need fast, automatic responses. But chronic stress keeps the brain in this mode, undermining the prefrontal cortex's regulatory functions. This may contribute to the impaired decision-making and increased impulsivity seen in chronic stress, depression, and anxiety. And it highlights the importance of stress management for maintaining optimal cognitive function. Brain imaging techniques allow scientists to observe brain activity in living humans. For most of neuroscience's history, studying the human brain required waiting for someone to die or suffer damage, then examining their brain post-mortem or testing what functions were impaired. Modern imaging techniques have revolutionized neuroscience by allowing non-invasive observation of brain structure and activity. Structural imaging techniques visualize brain anatomy. CT, computed tomography scans use X-rays from multiple angles to create cross-sectional images of the brain, useful for detecting structural abnormalities like tumors, bleeding, or skull fractures. CT is fast and widely available, making it valuable in emergency settings. MRI, magnetic resonance imaging, uses powerful magnetic fields and radio waves to create highly detailed images of brain tissue, with better resolution than CT and no radiation exposure. MRI can detect subtle structural differences and is used to diagnose conditions like multiple sclerosis, tumors, and stroke, and to investigate brain structure in research studies. Different MRI sequences provide different types of information. T1 weighted images highlight gray-white matter contrast, useful for visualizing brain structure. T2 weighted and flare images highlight abnormalities like inflammation, edema, or lesions. Diffusion tensor imaging, DTI, measures water diffusion in brain tissue, which is constrained by axon bundles, allowing visualization of white matter tracts. DTI can reveal disconnections between brain regions caused by injury or disease and has been used to study brain connectivity in development, aging, and psychiatric conditions. Functional imaging techniques measure brain activity. The most widely used is fMRI, functional magnetic resonance imaging, which detects changes in blood flow and oxygenation. When a brain region becomes active, it requires more oxygen, so blood flow to that region increases within a few seconds. This increase in oxygenated blood changes the magnetic properties of the tissue in a measurable way. By measuring blood oxygen levels, the bold signal, blood oxygen level dependent, fMRI can identify which brain regions are active during different tasks. A typical fMRI study has participants perform tasks or view stimuli while in the scanner, then compares brain activity during different conditions to see which regions are more active for one condition than another. fMRI has excellent spatial resolution by non-invasive standards, showing activity in regions just a few millimeters across, allowing researchers to identify specific brain areas involved in different processes. However, temporal resolution is limited. It can only detect changes occurring over a few seconds because it measures blood flow, which responds more slowly than neural activity itself. Despite this limitation, fMRI has revealed much about brain organization, mapping regions involved in perception, cognition, emotion, and action, and showing how activity differs in various neurological and psychiatric conditions. PET, positron emission tomography, involves injecting a radioactive tracer that accumulates in active brain regions, then detecting the emitted radiation. Different tracers can measure different things. Fluorodeoxyglucose, FDG, measures metabolic activity. Tracers binding to specific neurotransmitter receptors measure the distribution and availability of those receptors. Amyloid and tau tracers measure accumulation of proteins associated with Alzheimer's disease. PET provides information that fMRI can't, particularly about neurochemistry, but involves radiation exposure and requires specialized equipment to produce short-lived radioactive tracers, limiting its use compared to fMRI. EEG, electroencephalography, measures electrical activity by placing electrodes on the scalp. EEG detects the summed electrical activity of millions of neurons firing in synchrony in the cortex beneath. It has excellent temporal resolution, detecting changes in milliseconds, making it useful for studying the timing of cognitive processes, sleep stages, and epileptic activity. However, spatial resolution is poor. EEG can't pinpoint activity to specific small brain regions because the signal is smeared as it passes through tissue and skull. EEG reveals oscillatory brain activity, rhythmic patterns of electrical activity at different frequencies that reflect different brain states. Delta waves 14Hz occur during deep sleep. Theta waves 48Hz occur during drowsiness and some memory processes. Alpha waves 813Hz occur during relaxed wakefulness. Beta waves 1330Hz occur during alert, focused states. Gamma waves 3100Hz occur during active processing and attention. Magnetoencephalography measures the magnetic fields produced by neuronal activity. It has better spatial resolution than EEG while retaining excellent temporal resolution. But MEG equipment is extremely expensive, millions of dollars, and requires specialized shielding to block external magnetic fields, limiting its availability to major research centers. These imaging techniques have revealed much about how the brain works. Brain imaging studies have mapped the brain's organization, showing which regions are involved in vision, language, memory, emotion, and countless other functions. They've shown how brain activity differs in various psychiatric and neurological conditions, providing insights into disease mechanisms and potential treatments. They've allowed researchers to decode mental states, using patterns of brain activity to predict what someone is seeing, thinking about, or planning to do. Some studies have even decoded the content of dreams by identifying brain activity patterns associated with specific visual images and detecting those patterns during sleep. However, brain imaging also has limitations. fMRI shows correlations, which brain regions are active during a task, but doesn't prove those regions are necessary for the task. Activity could reflect computation essential to the task, or it could reflect incidental processes like attention, effort, or expectation. To determine necessity, other methods like lesion studies or brain stimulation are needed. Also, the brain is never inactive. There's always massive neural activity even during rest. And subtracting this baseline activity to isolate task-related activity involves assumptions that may not always hold. The default mode network, a set of regions more active during rest than during tasks, highlights this. This network is suppressed during focused tasks, but is active during mind-wandering and self-referential thought, showing that rest involves its own forms of processing. Another limitation is statistical challenges. With thousands of voxels, 3D pixels, measured in an fMRI scan, multiple comparisons must be carefully controlled to avoid false positives. Early neuroimaging studies sometimes used lenient statistical thresholds, leading to spurious findings, but methodological standards have improved. Recent concerns about reproducibility in neuroimaging have led to larger sample sizes, better statistical methods, and data sharing, improving the reliability of findings. Despite limitations, brain imaging has transformed neuroscience, providing a window into the living, thinking brain that was impossible just a few decades ago. Combined with other methods, patient studies, animal research, computational modeling, imaging contributes to a comprehensive understanding of brain function. Sleep is essential for brain function and involves distinct stages. Sleep is not a uniform state, but cycles through multiple stages with different brain activity patterns and different functions. Sleep is typically divided into non-REM sleep and REM sleep. Non-REM sleep has 3 stages. Stage 1, N1, is the lightest, a transition from waking to sleep lasting only a few minutes. Brain waves begin to slow from the alpha rhythms of relaxed waking to theta rhythms. Muscles relax, eye movements slow, and you become less aware of your surroundings. It's easy to wake someone from stage 1, and if awakened, they may not feel they were asleep at all. Hypnic jerks, sudden muscle twitches, sometimes occur during this transition, possibly reflecting neurons transitioning between waking and sleeping states. Stage 2, N2, is deeper, accounting for about 45–55% of total sleep time in adults. Brain waves continue slowing, interrupted by brief bursts of rapid activity called sleep spindles and larger waves called K-complexes. Sleep spindles, oscillations at 1216 Hz lasting a second or so, are thought to protect sleep from disturbances. They may suppress cortical responsiveness to external stimuli, helping maintain sleep. They're also involved in memory consolidation, particularly for declarative memories. K-complexes, sharp negative peaks followed by positive deflections, also suppress arousal and may play roles in memory processing and synaptic homeostasis. Stage 3, N3, is slow-wave sleep or deep sleep, characterized by very slow, high-amplitude brain waves called delta waves, less than 4 Hz. This is the deepest sleep stage. It's difficult to wake someone from stage 3, and if awakened, they typically feel groggy and disoriented. Slow-wave sleep is thought to be particularly important for physical restoration and memory consolidation. Growth hormone is released during slow-wave sleep, promoting tissue growth and repair. The immune system is strengthened. Studies show that people deprived of sleep are more susceptible to infections, and sleep enhances vaccine responses. Metabolic waste products that accumulate in the brain during waking are cleared more efficiently during sleep, particularly during slow-wave sleep, through the glymphatic system, channels surrounding blood vessels that allow cerebrospinal fluid to flush through brain tissue, carrying away waste products including beta-amyloid, which accumulates in Alzheimer's disease. This clearance function may partly explain why chronic sleep deprivation increases Alzheimer's risk. After progressing through stages 1–3, sleep doesn't simply continue in stage 3. Instead, it reverses back through stages 2 and 1, then enters REM, rapid eye movement sleep. In REM sleep, brain activity increases dramatically, with EEG patterns resembling waking activity. Eyes move rapidly beneath closed eyelids. These rapid eye movements can be observed and are one of the defining features of this stage. Breathing becomes irregular, heart rate and blood pressure fluctuate, and most skeletal muscles are paralyzed, preventing you from acting out dreams. This paralysis, called REM atonia, is controlled by the brainstem, and when it fails, people experience REM sleep behavior disorder, physically acting out dreams, sometimes violently, risking injury to themselves or bed partners. REM sleep is when most vivid narrative dreams occur, though dreams can occur in any sleep stage. REM dreams tend to be longer, more bizarre, more emotional, and more memorable than non-REM dreams, which tend to be more thought-like and less vivid. The function of dreams is debated. Theories range from dreams being meaningless byproducts of random neural activity, the activation synthesis hypothesis, to dreams serving important functions in memory consolidation, emotional processing, or problem-solving. Some evidence supports functional roles. REM sleep increases after learning tasks, particularly emotional or complex tasks. People awakened from REM sleep sometimes report insights about problems they'd been working on, and dream content often relates to recent experiences and concerns, suggesting dreams process waking experiences. Sleep progresses through these stages in roughly 90-minute cycles. The first sleep cycle has little REM sleep and more slow-wave sleep. As the night progresses, REM periods lengthen while slow-wave sleep decreases. By morning, most sleep is stage 2 and REM. This architecture suggests different stages serve different functions optimally at different times. Early night emphasizes physical restoration and consolidation of factual memories, while late night emphasizes REM-dependent processes like emotional regulation and procedural memory consolidation. The precise function of sleep remains debated despite decades of research, but it clearly serves multiple essential purposes. Sleep-deprived people show impaired attention, memory, decision-making, emotional regulation, and motor performance. After 24 hours without sleep, performance on many tasks is as impaired as if you were legally drunk. Reaction times slow, attention lapses increase, and errors multiply. Chronic sleep deprivation, even just getting 6 hours instead of 7-8 hours per night over extended periods, accumulates a sleep debt associated with increased risk for obesity. Sleep deprivation disrupts hormones regulating hunger and metabolism. Diabetes, sleep affects insulin sensitivity. Cardiovascular disease, sleep deprivation increases blood pressure and inflammation. Depression, bidirectional relationship. Sleep problems increase depression risk. And depression disrupts sleep and earlier mortality. Sleep also plays a crucial role in brain development. Infants and children sleep far more than adults, with newborns sleeping 16-17 hours per day in multiple bouts. REM sleep constitutes a much larger proportion of infant sleep, perhaps reflecting the brain's rapid development during these periods. As children age, total sleep time decreases and sleep architecture matures, with adult patterns emerging by adolescence. Sleep is regulated by two processes working together: homeostatic sleep drive and the circadian rhythm. Homeostatic sleep drive increases the longer you're awake, creating pressure to sleep that builds throughout the day. This drive reflects accumulation of adenosine, a metabolite that builds up during waking and promotes sleepiness. Caffeine works by blocking adenosine receptors, temporarily reducing the drive to sleep. The circadian rhythm is an internal approximately 24-hour clock that regulates the timing of sleep and wakefulness. This rhythm is generated by the suprachiasmatic nucleus in the hypothalamus, which contains neurons that oscillate with roughly 24-hour periodicity even without external cues. However, the rhythm is entrained by light. Photoreceptors in the retina, particularly melanopsin-containing ganglion cells, detect light and send signals to the suprachiasmatic nucleus, which adjusts the rhythm to match the external day-night cycle. The suprachiasmatic nucleus regulates production of melatonin by the pineal gland. Melatonin levels rise in the evening as light decreases and fall in the morning as light increases. Melatonin promotes sleepiness and helps consolidate the sleep period. Exposure to bright light, particularly blue light from screens, in the evening suppresses melatonin and shifts the circadian rhythm later, making it harder to fall asleep at your desired time. This is why screen use before bed often disrupts sleep. Conversely, morning light exposure helps advance the rhythm earlier, making it easier to wake up in the morning. These principles can be used to adjust to new time zones or shift work schedules. Strategic light exposure and melatonin can help shift the circadian rhythm to match new sleep-wake times. Consciousness is one of neuroscience's greatest unsolved mysteries. Consciousness is your subjective experience, the feeling of what it's like to be you. The redness of red, the painfulness of pain, the taste of chocolate, the feeling of being in love. These subjective experiences are called qualia, and they're central to consciousness. But how does the brain, a physical object made of cells and chemicals operating according to physical laws, generate subjective experience? How does electrical and chemical signaling among neurons produce the felt quality of experience? This is sometimes called the hard problem of consciousness, distinguished from easy problems, like explaining how the brain processes sensory information or controls behavior, which are challenging but seemingly solvable through standard neuroscience methods. The hard problem is explaining why physical processes give rise to subjective experience at all. Some philosophers argue the hard problem is unsolvable, that consciousness is fundamentally different from physical processes and can't be explained in physical terms. This view, called dualism, goes back to Descartes, who proposed that mind and body are separate substances. But dualism faces its own problems. If consciousness is non-physical, how does it interact with the physical brain? How does a non-physical mind cause physical neurons to fire? Every time you decide to move your arm and your arm moves, your mind seems to be causing physical changes. But how can non-physical substance affect physical substance? No mechanism for such interaction has been identified, and the idea seems to violate physical laws like conservation of energy. Most neuroscientists adopt physicalism, the view that consciousness is produced by physical processes in the brain, even if we don't yet understand how. Several theories attempt to explain consciousness in neural terms. Global workspace theory, proposed by Bernard Bars and developed further by Stanislas Dehaene and others, suggests that consciousness corresponds to information being broadcast widely across the cortex. At any moment, many brain regions process information unconsciously, visual features are extracted, sounds are analyzed, motor plans are prepared, but only information that's broadcast to a global workspace, a network of interconnected cortical areas, particularly in prefrontal and parietal regions, becomes conscious. This broadcasting allows different brain systems to access the information, enabling flexible, coordinated behavior. According to this theory, the difference between conscious and unconscious processing is not the type of processing, but whether information is locally confined or globally broadcast. Integrated information theory, proposed by Giulio Tononi, suggests consciousness corresponds to integrated information. A system is conscious to the degree that it integrates information in a unified way that can't be decomposed into independent parts. According to this theory, consciousness isn't all or nothing but exists in degrees, quantified as phi, a measure of integrated information. Even simple systems could have minimal consciousness if they integrate information, though most would have vanishingly small amounts. A thermostat, for example, integrates very little information and would have near zero consciousness, while the brain integrates vast amounts and has high consciousness. Importantly, this theory implies that very different systems, brains, computers, perhaps even simple networks, could be conscious if they have the right information integration properties, though creating artificial systems with high phi may be extremely difficult. Higher-order theories suggest consciousness requires not just processing information, but having thoughts about your mental states, metacognition. You're conscious of pain not just because your brain processes pain signals, but because you have thoughts about those signals, recognizing them as your own experiences. According to these theories, a system could process information without being conscious if it lacks the meta-representations, representations of its own representations, that constitute consciousness. This explains why we're not conscious of much of our brain's processing. We process visual features, language, motor control, but we're conscious only of those process contents that are additionally represented at higher levels as objects of attention or thought. These theories are still actively debated and tested. Brain imaging studies have identified neural correlates of consciousness, patterns of brain activity associated with conscious experience. For example, when people view a visual stimulus very briefly, sometimes they consciously see it, and sometimes they don't, even though the visual information reaches the brain either way. Comparing brain activity when people do versus don't consciously see the stimulus reveals that consciousness corresponds to sustained activity spreading from visual cortex to frontoparietal networks, consistent with global workspace theory. Anesthesia, which eliminates consciousness, disrupts communication between cortical regions without necessarily reducing overall activity levels, suggesting consciousness depends on specific patterns of connectivity rather than just activity level. Disorders of consciousness provide further insights. Coma is a state of complete unconsciousness where the person doesn't respond to stimuli and shows no sleep-wake cycles, typically lasting days to weeks. Vegetative state, now called unresponsive wakefulness syndrome, involves sleep-wake cycles and reflexive responses, but no signs of awareness. Patients' eyes may open and close, but there's no evidence they're conscious. Minimally conscious state involves inconsistent but definite signs of awareness, following commands sometimes, responding to stimuli purposefully occasionally. Locked-in syndrome is distinct. Patients are fully conscious but almost completely paralyzed, unable to move except sometimes eye movements, illustrating that consciousness and motor function can be completely dissociated. These states show that wakefulness and consciousness can be dissociated. You can be awake without being conscious. Studies using brain imaging have revealed that some patients diagnosed as vegetative may actually be conscious. When asked to imagine playing tennis or walking through their house, these patients showed the same patterns of brain activity as healthy people performing those imagery tasks, suggesting they were conscious and able to follow instructions despite being unable to move or communicate. These discoveries have profound implications for medical care and ethical decisions about life support. Some patients previously thought unconscious may actually be aware, experiencing their situation but unable to communicate, a terrifying prospect. Understanding consciousness is not just a philosophical puzzle. It has practical importance for medicine, determining who is conscious and in need of pain management, law determining who is capable of decision-making or culpable for actions, ethics determining the moral status of patients, fetuses, or animals, and potentially artificial intelligence, determining if machines could be conscious and what that would mean. While we've learned much about the neural basis of consciousness through brain imaging, patient studies, and theoretical work, the fundamental question, why physical processes give rise to subjective experience, remains open, one of the deepest mysteries in science and perhaps the deepest mystery about human existence. We've traveled far tonight, from the neurons firing in patterns that create your thoughts and memories, forming connections that strengthen with experience, to the attachment bonds that shape emotional development and influence relationships throughout life. From the biases systematically distorting thinking, but that you rely on thousands of times per day, to the sleep stages cycling every 90 minutes, each serving different functions for body and mind. From neurotransmitters crossing synapses in milliseconds to personality traits showing remarkable stability across decades. From the ways authority and situations shape behavior in unexpected directions to the resilience allowing adaptation and recovery from adversity. But here's the thing, psychology and neuroscience aren't separate from you. This isn't abstract knowledge about other people's minds or brains in general. This is about your mind, your brain. Right now, as you're processing these words, working memory is holding information in conscious awareness. Attention is filtering sensory input. Schemas are providing frameworks for understanding, and countless unconscious processes are happening beneath awareness, regulating breathing and heartbeat, processing peripheral vision, monitoring body state, retrieving relevant memories without conscious effort. The biases we discussed, you experience them constantly. The attention limitations. They constrain your perception right now. The power of situations to shape behavior. You're influenced by context just as everyone else is, even though it feels like your behavior comes entirely from autonomous choice. The placebo effect. Your brain can produce physiological changes based on expectations, demonstrating that mind and body aren't separate domains. The social influences. Countless aspects of how you think and act have been shaped by culture, groups, norms so internalized you don't notice them. Understanding your brain provides insight into yourself and others. When you catch yourself in a cognitive bias, noticing you're seeking information confirming what you already believe, or making a decision based on an arbitrary anchor, you can pause and compensate. When you understand that attention is limited and multitasking impairs performance, you can arrange your environment to minimize distractions when focus is needed. When you understand that memories are reconstructed rather than recorded perfectly, you can be appropriately skeptical of vivid recollections, especially in consequential situations. Understanding the brain also cultivates compassion. When you recognize that behavior is influenced by situations and neural states, not just character, you can be less quick to judge others and yourself. The person who seems rude might be stressed or distracted. The colleague who made a terrible decision might have been operating under time pressure and limited information, with prefrontal cortex function impaired by stress. The weakness you see in yourself might be a normal human limitation that everyone shares, but most people don't discuss. Psychological and neuroscience research consistently shows that people underestimate situational influences on others' behavior, while overestimating situational influences on their own behavior. Understanding these patterns helps you see others more generously and hold yourself appropriately accountable. Your brain also illuminates possibilities. Neuroplasticity means your brain can change throughout life. Learning physically alters your brain, strengthening connections, representing new knowledge and skills. Therapy can be effective, helping develop new patterns of thinking and behaving that improve mental health and functioning. Interventions can promote resilience even after adversity. You're not fixed at birth or even after childhood. Change remains possible, though it requires effort and often support. Understanding this can inspire hope while avoiding naive optimism that change is easy. Finally, understanding your brain reminds you that you're not alone. The experiences you have, anxiety, sadness, joy, confusion, the sense that you're fundamentally different from everyone else, are human universals. Everyone experiences cognitive biases, memory failures, attention lapses, and emotional ups and downs. Everyone has an inner experience that feels unique, but is built from the same neural architecture and psychological processes. The details differ, your specific memories, thoughts, personality, but the underlying mechanisms are shared. You're one instance of the human mind, unique in particulars but similar in fundamentals to the billions of other minds that have existed and will exist. If you're still awake, there are more videos waiting. But if sleep found you somewhere along the way, then this worked exactly as it should. Rest well, knowing that as you sleep, your brain continues its essential work, consolidating memories from today, processing emotions, reorganizing neural connections, and preparing for tomorrow. Sleep itself is a psychological and neural process, one we still don't fully understand, but one that's absolutely essential to being human. Your mind, even in sleep, is never truly at rest. It's simply engaged in different work, the quiet maintenance and reorganization that makes consciousness possible when you wake. The neurons continue firing, maintaining their connections, adjusting their strengths, preparing the circuits that will produce your thoughts, feelings, and experiences tomorrow. Your brain never stops, from first moment to last, generating the continuous stream of experience that is your life, the flow of consciousness that makes you you. See you next time.